The overall goal of the following protocol is to generate prostate cancer patient-derived xenografts from circulating tumor cells. This is achieved by first collecting peripheral blood from patients with advanced prostate cancer. As a second step, the blood mononuclear cell compartment is isolated, which contains the circulating tumor cells. Next, the mononuclear cells are stained with a CD45 FITC antibody in order to select the circulating tumor cells using flow cytometry. The cells are then injected into immunocompromised mice. Results show the generation of prostate cancer xenografts based on the injection of circulating tumor cells isolated by flow cytometry from the peripheral blood of prostate cancer patients.
This method can help answer key questions in the prostate cancer field by generating new experimental models that can be used for the molecular characterization of prostate cancer and the development of new biomarkers. Demonstrating the procedure will be Estrelania Williams, a student from the laboratory, and Uma Chippada, a researcher from the Oncology department here at Mount Sinai.
To begin, collect whole blood from select patients with metastatic prostate cancer, as they have the potential to have high numbers of circulating tumor cells in their peripheral blood. Using a 10 milliliter serological pipette, transfer the whole blood into a 50 milliliter polystyrene conical tube along with Hank's balanced salt solution in a 1:1 ratio. Gently pipette the mixture to homogenize it.